Next topic to remember is DNA replication. Okay. First of all, occur in 5 to 3 direction. Okay. Occur in 5 to 3 direction in continuous and discontinuous fashion. Okay. Here you can see that DNA. Okay. First it will be unzipped, and then you will have continuous formation of DNA, and this is a uncontinuous fashion of DNA synthesis. We will discuss that more complex and eukaryote and prokaryote is semi-conservative, okay, but share analogous inside. First of all is the origin of replication. It doesn't mean replication will start from anywhere. There are certain point from which replication will start. That point are a particular consensus sequence in genome where DNA replication begins. Okay, this MCQ have been asked in Jupyter in same way. Okay, it can maybe single in prokaryote, multiple in eukaryote. So DNA replication will be occur at a single level in prokaryote, but in eukaryote it will be at multiple level. From where, okay, region of replication start, it is the Tata box region. Okay, okay. 80 race sequence that is called Tata box region are found in promoter and origin of replication. They are mainly found in promoters and origin of replication. That's a Tata box. When you start new life, okay, origin of new life, first you have to say Tata. Tata, no, bye bye. Okay, then you will start the new life, origin of new life. So this is the way I have framed this point. Tata, you know that my local language word they use for bye bye, okay, and then you'll start the new life. So this is the way to cram that. Okay, then is a replication code. Okay, replication code here you can see it is like look like Y. Okay, here you can see that it look like Y. Okay, here you can see, guys, it is look like Y. So this is a replication code. So, okay, what do you mean by semi-conservative? Okay, so for semi-conservative, okay, suppose this is a DNA, it will be unzipped now. This is parent DNA. Okay, new DNA will be okay. So next I'm drawing this is new DNA that form red one. Okay, so it is semi-conservative. 50% this will be from parent DNA and 50% will be new form. It is semi-conservative. Okay, so conservative like old, semi is half. So here, okay, is that right? Okay, this DNA will be half from parent and half will be from the you can say okay that is done. Here for replication for okay, here Y shape will form. Okay, so next is helicase. Helicase unwind DNA template at replication form. They unwind, like can say unzip. Okay, the DNA. Otherwise, DNA is double standard. Okay, so here you can see that unzipping. This is zipped. This is unzipped. It is done by helicase. Here you can see this is helicase. Unzipping the DNA. Okay, helicase halves the DNA. Deficient Bloom syndrome. Okay, so okay, VLM gene. Okay, so what is Bloom syndrome? Guys, when you will have issue of helicase, okay, you will have a disease is called. What do you mean by that disease? That disease is called progria, early aging. Progria, you will have skin issues. Okay, you will have early skin cancer. You will have photosensitivity. Okay, so that is Bloom syndrome. Okay, that is a that is a you can say that is a Bloom syndrome. Okay, progria early aging. Okay, you will have early aging, and then you will have skin. Okay, skin issues. You will have skin cancer. Increased risk of skin cancer. You will have photosensitivity. So that is Bloom syndrome. Okay. How I have crime? Okay, that is for my local language student. Halkat Jawani. Okay, Helicase Jawani like bloom, bluer. You have to remember what bluer. In this way, I have crammed the word. This is main point, progria, early aging. Okay, you will have early bluer, blooming. Helicase Halkat Jawani. That is a word. Okay, so progria is IGD state. You will bloom earlier. That is also point. Okay. So like if blooming occur at, for example, occur at 20 years. In this case, blooming occur at half of that age. So half by half, I have make mnemonic for foreign student. Half is highly clear. So blooming will occur at half of age. So this is for foreign student. So remember, Bloom syndrome is important, not given in book. The early progria, not early progria mean early aging. Okay, a child will be look like older adult. Okay, a classic. Okay, it's okay. Uh, what do you say? Okay, in Harry Potter, a classic was classic. Kid was given. Okay, that with long hair in Harry Potter. That was God was having progria. Okay, you can Google it too. Then is single stranded binding protein. When helicates have, here you can see when helicates have separated these strands. Okay, so then you will have single stranded binding protein. Okay, here you can see I am drawing. It is given. These are single stranded binding protein. Here you can see the single stranded binding protein. What are the functions of them? There are two functions. What is they prevent from re-annealing. One strand is open, that strand can again, you have unzipped the strand, it can be zip up again. Okay, to from re-annealing, recombine it. Okay, you can have single stranded binding protein. That single stranded binding protein prevent from re-annealing. Second important point, that is high yield. Degradation by nucleases. Single stranded binding, okay, single strand is prone to damage by nucleases. The single person are prone to damage by, proteases, nucleases. So these are the function of single strand binding protein. Okay, degradation by nucleases. Remember, prevent re-annealing, degradation by nucleases. So that is single stranded binding protein. DNA topoisomerase. Okay, here you can see whenever you will have, okay, so when you will have unzipping of this replication force, okay, in the forward region, okay, here you will have hypercoiling. Coilation will increase in the forward region and coiling will decrease in the backward region. Actually, that is bad one. This is called underwinding. This is called overwinding. Overcoiling, less coiling. Both are bad one. Okay, important. Remember the concept of that. Okay, in the next coming region, you will have hypercoiling. Okay, so to prevent that is a topoisomerase. Okay, here you can see topoisomerase. Okay, they remove supercoil as needed due to underwinding and overwinding. Okay, they will remove supercoil. So there are different topoisomerase. One create one nick, one break, and topoisomerase two cause double nick. They cause two stranded breaks. The single nick and double nick. That is different between topoisomerase one and two. And use your, okay, irinotecan, topotecan. Word is ending at one. Irinotecan and topotecan, giving a sound of an. Okay, they will block topoisomerase one. A topocyte, tinopocyte, topo. That is giving sound of two. Okay, it will inhibit topoisomerase two. And this is very high yield MCQ. Fluoroquinolone will inhibit topoisomerase two and topoisomerase four. So fluoroquinolone for four. I have remembered this. Fluoroquinolone four. 4 by 2, half is 2. Okay, so fluoroquinolone will inhibit topoisomerase 2 and that is called DNA gyrase. Examiner can ask. Okay, and topoisomerase 4. Okay, then is the primase. Make a RNA primer on which DNA polymerase can initiate replication. Replication cannot start without primer. Why? Because DNA polymerase is the initiator of that make the DNA, new DNA. But it needs some base. Here you will have RNA primer. So here you will have RNA. Okay, and that last segment will provide OS group. Okay. Then DNA polymerase will come, use this third and OS group and elongate the next DNA. So here is line, easy line. Okay, RNA primer is required for initiation of replication. Okay, DNA polymerase III prokaryote only elongate leading strand by adding deoxynucleotide to the third and elongate lagging strand until it reach the primer of preceding fragment. So that is just adding the new nucleotide. DNA polymerase has 5 to 3 synthesis. Guys, remember, transcription, translation, replication always start from 5 to 3. Okay, synthesis from 5 to 3. This is MCU. Proof we read with 3 to 5. Making the DNA in 5 to 3 direction and proofreading the DNA to 3 to 5 exonuclease. If in the 3 to 5 is like backward. If the backward there is a issue, there is a wrong DNA nucleotide is added, it will have proofread that area and cleave that area with exonuclease. Remove that nucleotide. So that is the proofreading function. Drug blocking the DNA replication often have modified 3 OS group. Okay, thereby preventing the addition of next nucleotide. Here I have told you the OH group. OH group is necessary for successive nucleotide. Here you can see this one nucleotide provide OH group. Okay, then next nucleotide. Okay, so this OH group is necessary for binding one nucleotide to other nucleotide. Okay. If you have modified, for example, in this side I have add CH3 group here, or I have add any group. So this is now modified nucleotide. So now you cannot add the next nucleotide. So this is called chain termination. DNA polymerase 1 is present procured. Degrade RNA primer. Replace with DNA. We have discussed that RNA primer is used for DNA replication, initiation of DNA replication. When the replication started, you don't need RNA primer in DNA. So you will remove that RNA and replace with DNA. Here you can see same function as that, also excise RNA primer with 5 to 3 exonuclease. Here see the difference, see the MCU, see the important point. DNA polymerase 1 have 5 to 3 exonuclease activity, DNA polymerase 3 have 3 to 5 exonuclease activity. See the difference, combine them. Okay, don't confuse. DNA ligase, catalyze the formation of phosphodiester bond. It link the DNA. When one DNA is formed, for example, this DNA is formed, this DNA is formed, combined with ligase. Next last topic is telomerase, very high-yield topic. It is present in eukaryote only. It is a reverse transcriptase that add DNA to third end of chromosome to avoid loss of genetic material with every duplication. So here I am drawing. This is you can say this is first DNA. Then next replication will occur. It will be less. It will be like that. Less replication. Coming less replication. Length will be decreased. Chromosome length is decreasing. Length is decreasing like that. With successive replication, with successive duplication, length of chromosome will decrease. Then a critical length will occur. At that length, suppose this is a length, you can't replicate the DNA. This length is this length is called critical length. After that length, if decreased length occur, you can't do DNA replication. So that's why nature has given you telomerase. Telomerase actually extend here I have extended the length of DNA. Chromosome have been shorter, DNA have been shortened. Then telomerase will come, add more nucleotide. So here you will have replication potential will be regenerated now. Now it can be replicated. That is the reason. So there are two things given in telomerase. One is the DNA polymerase. Other is this DNA segment. Mnemonic is telomerase tags for greatness and glory. TTAGG to third and point of a chromosome.